The broadcast is now starting. All attendees are in listen-only mode.
Hello, everyone. Welcome, and thank you for attending our 5th session for the EMN live webinar series. My name is Michelle Reed. I am currently a scientist at Crystal Therapeutics in Maastricht, Netherlands. And on behalf of the Early Member Network Committee of the Metabolomics Society, I will be co-hosting this event with Peruva Kulkarni, a postdoctoral researcher at Radboud University Medical Center in the Netherlands. Thank you again for joining us. If you would like to hear this talk at a later time, the webinar will be uploaded to the Metabolomics Society website with a link distributed through our social media networks. We encourage you to connect with us online via Facebook and Twitter for future content. And feel free to email us with suggestions for speakers or topics and other input. Today we have 2 talks. The first from Professor Tim Ebbels at Imperial College London. His talk is entitled Computational Metabolomics. What is it and why do we need it? Professor Tim obtained his PhD in astrophysics from the University of Cambridge, and in 1998 moved into bioinformatics via postdoctoral work at Imperial College. His group focuses on application of bioinformatics, machine learning, and chemometric techniques on post-genomic data, with a particular emphasis on computational metabolomics. His work focuses on modeling the analytical technologies used to obtain metabolic data, as well as data integration, visualization, network analysis, time series, and metabolite annotation. He is particularly known for the Batman software for analyzing complex metabolic NMR spectra. Tim is an active member of the Metabolomics community, having served as the director of the International Metabolomics Society from 2012 to 2018, and during this time as secretary from 2014 to 2016. He's also co-founder of the London Metabolomics Network and is an editorial board member for BMC Bioinformatics and the Journal of Chemometrics. He has a strong commitment to postgraduate education, serving as director of MRes in biomedical research at Imperial College, and has trained over 700 students in this role. Our second talk is from Cecilia Vida at Imperial College London. Her talk is entitled Pathway Analysis in Metabolomics, Recommendations for the Use of Overrepresentation Analysis. Cecilia is a bioinformatics doctoral student supervised by Professor Tim Ebbels and Dr. Rachel Lay at Imperial College London. Her research focuses on developing methods for the integrative analysis of multi-omics data at the pathway level, with a particular emphasis on pathway analysis methods for metabolomics data. Her overarching aim is to leverage biological pathway information in the integration of multi-omics datasets in order to identify robust cross-omics molecular signatures, as well as enhance predictive model performance and interpretability. Before our talks, I'd like to announce that the last 2021 webinar will be on December 13th. Dr. Julian Bouchard and Miguel de Figueiredo will discuss data structures as well as key knowledge discovery in metabolomics and a strategy to handle high-dimensional metabolomic data from unbalanced multifactorial designs. For today's talk, please remember to submit questions throughout the presentation in the questions tab, and include who the question is directed towards by indicating the speaker's first name followed by your question. I will now give the presenter control to Professor Ebbels, and the platform is yours to begin.
Right. I think I might have displayed the wrong screen. Let me just try again. Okay, can you see my slides?
Yes.
Okay. They should be on presenter view now. Can you see that?
Yes.
Fantastic. Okay, so thanks to the EMN and the Metabolomics Society for inviting me to give this presentation. It's always a pleasure to speak to members of the Metabolomics Society and all the metabolomics people around the world, whether it be in person at the conferences or virtually like we're doing here. So what I'm going to try to do over the next 20 minutes is to give you a bit of an overview of what I see as computational metabolomics. That's a term which has come into use over the past sort of 4 or 5 years, I think, and it's an area which I sort of find myself as actually being my main area of work. I probably wouldn't have used that term 10 years ago, but it's actually something that's come along and is a nice way to describe what I do. So if I want to describe what I think it is, I guess computational metabolomics could be defined as computational, statistical, machine learning and AI techniques applied to data, problems and questions in metabolomics. So a pretty broad definition there. Essentially, if you've got metabolic data, so metabolomic data as represented down here on the left with a sort of table of metabolites, abundances perhaps, and you're doing some kind of statistics on it, usually with some kind of computational algorithm, then you're doing computational metabolomics. So it can be anything to do with metabolic data as long as you're applying some kind of statistical or computational analysis. Why do we need computational metabolomics? Well, I'm hoping I don't have to persuade too many people of that, but essentially metabolomics has always had a strong computational or statistical or data analytical component. So you can see on the right here, I've got a little Venn diagram. I mean, often people have presented this as metabolomics is being kind of the fusion of 3 different things. You know, the biological aspects, obviously, metabolism, key part of biology, chemistry, both the sort of biochemical aspect, but also the analytical side of you know, the instrumentation we use to obtain the measurements, and then the informatics or mathematical statistical side, which has always been there really from the beginning because the data are quite large and complex. But why do we need computational metabolomics more so now than we did before? Well, essentially we've got more data. So I put N there. That means really we're getting larger and larger data sets. Those of you involved in epidemiological research will be very familiar with data sets of thousands of individuals, perhaps even more so. And there are data sets coming along, for instance, like the UK Biobank, where we have upwards of 500,000 people. The data itself is more complex. So we're getting data which has a more complex structure, whether that be in terms of the raw measurements, you know, the structure of the chemical information that comes from the mass spectrometer or from the NMR spectrometer, for instance. But essentially part of that is that there are more and different assays being applied. So say 10, 15, 20 years ago, we might have had a typical metabolomics experiment, just applying a single assay to a single smallish set of samples. Nowadays, it's quite common for us to apply several different metabolomic assays to the same samples to broaden the coverage of the metabolome. One key problem, as most of you will know, is that many metabolites are unknown, or many signals in the data. We don't know what metabolites those correspond to, and we get more and more of those as our instrumentation gets better. So if you can think as the sort of detection thresholds and lower limits of detection get better and better, we're going to see more and more of the metabolome, which is great, but we'll have many, many more unknowns to identify. The experimental designs are becoming more and more complex. Used to be that the typical study was simply a sort of case control or treated versus control kind of 2-group comparison. But nowadays in the literature, you can see many more different designs, including time series and nested cohort designs and things like that. And finally, of course, many of us are not just using metabolomics, but we're applying lots of different types of biological assays and those from other types of omics. And we want to combine those often with our metabolic data to tell the sort of whole biological story. So all these aspects make the need for computational methods in metabolomics more and more acute. So where does computational metabolomics fit into the sort of classic workflow that we use in metabolomics? Well, this is my metabolomics workflow slide. If we start off with a biological question up at the top here on the left, we'll have to do some experimental design, of course. You know, that includes both the biological experiment, you know, how do we treat our samples, how do we induce the effect that we want to see. We'll have to actually obtain some biological samples, whether that be a biofluid or an extract or something like that. Then there'll be some sample preparation. So how do we prepare those samples so that they can actually be analyzed by the instrumentation that we have. Then we go into the key part, which is the data acquisition. So we're actually on the instrument and we get some lovely squiggly data you can see there. And then on the right hand side, you can see lots of data aspects. So we have some pre-processing, which hopefully will end up in a nice table of concentrations or something like that for us. There'll be some generic data analysis, for want of a better word, which could include statistical tests. It could include multivariate modeling or regression or something like that. But there's often a step which is metabolite identification. And this is really the key point of the untargeted analysis, where you're covering as many metabolites as you can. You often don't know what all those signals in the raw data represent. And so there are many computational aspects that can be applied in the metabolite identification stage. And finally, we have some kind of biological interpretation stage where we want to actually find what's the biological story that this experiment told us. The colors here are supposed to represent, you know, green for sort of biological knowledge, orange for sort of analytical chemistry knowledge, and blue for more data analytical knowledge. And that's where you can see there's a fusion of these different things that are required. And the computational metabolomics really comes into these main stages that I've highlighted in yellow here. So definitely into the experimental design, because you want to make sure that statistical aspects, you know, for instance, such as power, have been addressed so that you've got enough statistical power to test the question that you want to answer. And then on this right-hand side, as I said, pretty much all of those are very computationally heavy these days. And as I pointed out with this arrow, the metabolite identification conundrum is a really fundamental problem for metabolomics, and particularly computational metabolomics. And the fact that we can't immediately identify all the compounds that we're assaying really affects many, many different stages of the pipeline. So what different fields are there in computational metabolomics? I thought I would just show you a few of them in a sort of kind of representation of a map. So here we have a map with a sort of old picture of the world on it. But what have we got here? Well, as I said, experimental design is a big factor, and that needs to go in right at the beginning of any kind of workflow for putting a metabolomics experiment together. So it could be, as I said, statistical power, but it also relates to the sampling design. So how do you decide who and when you're going to sample in a human study, for instance? It also goes, of course, into the analytical design or experimental design. So how do you decide on the run order of your samples? Or other aspects such as the QC, the quality control. It also is clearly a key part of pre-processing of data. So once the data starts to come off the instrument, essentially what we've got are simply ion counts from a mass spectrometer or intensities in the NMR world. And there are many different signal processing algorithms that can be applied, and a whole pipeline of techniques which need to be applied in order to transform essentially raw spectroscopic or spectrometric data into something which is amenable to statistical analysis to answer the biological question. One aspect of this which relates strongly to computational metabolomics itself, if we're developing new methodological techniques, we quite often want to simulate data. And it's actually quite hard to simulate realistic metabolomic data that would look similar to what you would get from a real instrument. Down here you can see is one of our pictures from many years ago where we put together some software for simulating biological NMR spectra. But this is an area which I think is a bit underdeveloped. It's not easy to do those simulations. As I mentioned, annotation and identification of unknowns is a really big area of computational metabolomics, and I'm going to talk about that a bit later on. Of course, we have all the usual machine learning and multivariate statistical approaches for producing exploratory and statistical predictive models. So for instance, can we classify groups that we know to be existent in the data? Can we see trends with respect to some kind of continuous external outcomes such as age or body mass index or something like that? We also have these more complicated designs. So time series is one aspect of that. So here you can see two different groups, two different treatment groups followed over time. And there's a really complicated trajectory over time. And you know, how can we follow that and model that with traditional tools? It's not actually very easy. Traditional time series methods can usually require many, many data points over the time domain, and that's not something you usually have in a metabolomics experiment. So we do need some new methods for those sort of things. Visualization of the data is a really key point as well. Don't forget, this is multi-dimensional data, often not just many different metabolites measured, but possibly in different assays and possibly integrating with other things. So how do we visualize this really complicated data is a key point. And one of the ways we can do that visualization is via a network approach. So there are many different applications of sort of network science in different areas in computational metabolomics. Here's one which is just showing some statistical associations between, in this case, genomic SNPs and metabolite levels. Pathways is a key point for interpreting the data towards the end of the pipeline, and that's something that Cecilia is going to talk about later on. And finally, a big growth area in computational metabolomics is integration of data. So when you have data from multiple different assays or multiple omics, you want to be able to put those together in some kind of coherent view. So as I said, I'll be focusing on these two parts, but don't forget that there are plenty of other areas of computational metabolomics which I'm not speaking about just now. So let's talk about metabolite annotation as a focus area. So essentially what we're trying to do here is relate our spectroscopic data, or spectrometric data, to some kind of metabolite structure. So how do we get from this domain, which is essentially a spectrum, to something that is a chemical structure? It's actually quite a difficult problem in terms of sort of computational science. This would be the standard workflow for doing that, I think, in conventional LCMS, so liquid chromatography mass spectrometry. And most of you will be familiar with this, I'm sure. If we have an unknown, we will probably get that from a list of interesting mass to charge ratios or retention times that come out of some statistical analysis of data. We'll often do some kind of database search, okay? So we might use the accurate mass, the MZ. That might generate a formula, and we can search by mass in a very generic metabolite database, things like HMDB, for instance, or MassBank or something. We can also search by formula for that, but often we want to use more parameters to narrow down the possible metabolite identities. And the retention time is a really important piece of information, and you can do some searches by that. However, the problem with that is that it's not a universal parameter. The retention time on any given chromatographic system depends on lots of different factors of how the system is set up. And so it's hard to produce a generic database where you can search that parameter to identify things. So that'll often be done on a local database within a given lab. And of course, in mass spec, we have fragmentation approaches. So we can do MS2 or MSMS, where we break up the compound into its constituent parts and we look at the spectra of those, and we can search MSMS databases. So hopefully out of that, we come up with some candidates, and that gives us a so-called annotation. And don't forget, annotation is putative identification. It's identifying candidate structures, but it's not the final story. It's not 100% confident identification. So what can we do to improve on that kind of simple database searching approach? We can do things like grouping peaks together. So instead of just looking at one mass to charge ratio or one set of data, we can group things together using the fact that we observe in mass spectrometry different isotopologues, we observe adducts, neutral losses, different fragments and multimers and things like that. And people have shown that if you do link the peaks together in this kind of way, you can reduce the number of features that need to be identified. And not only that, if you have a group of features that you know to be related or you think are related from the same compound, then it allows you to search the databases in a much more specific way. Another way to group peaks together, of course, and this is well known, is to use correlations. That's correlations of the intensities of those ions either across the peak shape. So here you can see two peaks that are co-eluting from the same molecule. So it might be two different adducts, for instance. Here you can see two peaks that are not co-eluting, probably from different molecules. You can use that correlation, or you can use the peak abundance. So this is looking at the area under the peak in different, for the two different peaks across your range of samples. And if they're highly correlated, there's a good chance those might be related. So we can put those together, and this is something we've done in our Metabolomics software, which we hope to soon be published in Analytical Chemistry. Just briefly to go through this, it's a simple pipeline where we combine compound libraries and unknown feature lists. We use this peak shape correlation. We do some isotope checks, and then we check against the compound library to see whether the pseudo spectrum that we pulled out using these correlation approaches matches any of the fragmentation that we see in the compound libraries. And using a matching approach and scoring, we can then get a list of compounds essentially that could be candidates. And this works fairly well. It's not perfect, but you can get a good percentage, say 70 or 80% of the peaks sort of ID'd in this way, and the sort of validity of it is pretty good. Again, it's quite a high percentage of good hits. There's plenty of other things you can do, of course, though. An obvious one that's been around now for quite a long time is doing some de novo or in silico fragmentation. So there you take your compounds in your database of known metabolites, and you use some computational chemistry to fragment those inside the computer. And what you can do is build fragmentation trees. So taking the whole compound, breaking bonds essentially, and producing smaller and smaller fragments, which you can then calculate MS2 spectra for. And using those predicted spectra, you can match those to what you see in your data to try and see if you've got that compound in your data. But of course, it's not easy to do, partly because the computational chemistry is difficult, although that's getting better, but also because really to get high specificity for this, it'd be nice to do multiple fragmentations, and typically we often only are limited to MS2, so one level of fragmentation in our data. Another approach which I think is really nice and perhaps isn't used widely enough is using biotransformations. So David Wishart's group has published some tools for this, for instance. We can use essentially already known biochemical reactions to predict what types of metabolism will be going on in any given organism, so we can predict how different compounds will be metabolized. And in the reverse sense of that, we can say if we've got an unknown, could that be a transformation product of a given starting compound? So using a whole series of different enzymatic reactions, we can see whether the network of predicted compounds corresponds to any of our unknowns in the data. And here's just one sort of latest result from the group here at Imperial. This is work from Rui Pinto. And what he's done is used essentially a correlation approach, but this time using UMAP. So that's a relatively new dimension reduction technique from machine learning. What he does is use the similarity of the abundances of different features. So these are coming from an LC-MS assay across thousands of different individuals in many different studies, and use that to cluster the features together. And what you're seeing here is the different clusters produced by the UMAP study. The colored features here are the ones which are already identified. So we have things like lyso-PCs, lyso-PEs, acyl carnitines, etc. And then the black features are the ones which are unidentified. And you can see that there's a strong clustering to do, which is related to the chemical class. You can see, for instance, down here, we've got triglycerides and diglycerides, for instance. But you can also see that there's plenty of, as yet, unidentified features here. And of course, the fact that they're clustering together is a good suggestion that those might also be triglycerides or diglycerides. So this seems to be a pretty promising way of looking at identifying unknowns using a simple association, sort of statistical association point of view. So what are the issues with annotation? Well, it's the usual stuff. We have plenty of unknowns, but they come in different types. We have unknowns which we expect to be there, so things which we haven't identified in the sample but they're well-known molecules. They exist in databases. We also have the unknown unknowns, the so-called dark metabolomes. So the metabolites which are actually not known in current databases, haven't been identified before, haven't made it into a database. How can we identify those compounds, especially in some areas which may be less prevalent in the databases, like plant metabolites, for instance? Of course, chemical space itself is absolutely vast. So if you think of just glutamine, so simple amino acids, there are more than 1 million structures corresponding to the chemical formula for glutamine. So if you think of trying to transform, you can get the formula, of course, from the accurate mass reasonably easily, but trying to work out which of these many structures actually corresponds to your compound is very difficult. And it gets progressively worse. You can see the sort of exponential increase in the number of different structures that can correspond to it as you go along in larger and larger masses. Another big problem is that we don't have fragmentation spectra for all our unknowns. We only tend to have those for the most abundant peaks in the data. And as I said before, it's quite difficult to transfer or predict the retention time in a generic way corresponding to different chromatographic systems. And finally, it's quite difficult to estimate the confidence in our identifications. So depending on which database we use, what algorithm we use, the confidence in those computationally derived annotations will be different. But there are prospects for the future, so there are plenty of interesting aspects. One of them is the deep learning, so the sort of AI and deep learning things that you will have heard from computer science and elsewhere. One thing that's very good at is avoiding the so-called feature engineering thing. So it's quite good at mapping very different domains together. And as I mentioned at the beginning, spectra are a very, very different representation of data to the sort of chemical structures. But deep learning could have the potential to relate those two domains in a very unique way. Another way which I think is really important coming up is substructure learning. So of course, chemical compounds are made up of many different substructures. And of course, we may have seen many of these substructures before in our databases, even if the compound we're looking at, which is a composite of those substructures, is not known. And so methods like MS2LDA, for instance, from Justin van der Hooven and collaborators, are a really good way of looking for those substructures and trying to do some annotation using them. And finally, I think it's important to think about what information we really need in order to make a confident identification. So we know we have things like mass to charge ratio and retention time, and perhaps some fragmentation spectra in mass spectrometry, but how much information do we really need to make that identification confident? There may be cases where, even given a very large number of different types of assay data, we still can't be confident about an annotation. Okay, so the second topic I wanted to cover here, and I'll try to make this fairly quick, is data integration. So essentially, if you have two different data sets or more than two different data sets corresponding to your problem, what do you do in terms of putting them together? I would always like to think of this in terms of two different dimensions. So I call them horizontal and vertical data integration. For me, horizontal integration is when you're combining multiple assays. So we have a set of individuals here. Each row of these matrices is an individual. And then we've got our different assays. So it might be NMR and mass spec and other aspects here, or it might be several different mass spectrometry assays. And we often try to relate that to some kind of outcome, by which I mean the different biological classes or some other relevant phenotype, for instance. So that's what I would call horizontal integration. And the other aspect, the other way of doing it, is vertical integration. So this would be where you have the same assay, so it might be the same LCMS, I don't know, a lipid assay or something, and you're looking at combining that data from different studies together. So it might be you have a set of individuals from study 1 and a set of individuals from study 2, and you want to combine that in some kind of joint model. How do you do that? I should note that there's different definitions of horizontal and vertical integration in the literature, so don't be surprised if you see people referring to it the other way around, especially people from the sort of gene expression world. But vertical integration has some very specific challenges in terms of, to do this, you've got to have a reproducible assay, you've got to believe that the biology going on in the different data sets is kind of similar or comparable at least. And one of the really key problems is for untargeted metabolomics. How do you match a variable here, which may be only identified by mass and retention time, to a variable here, which is also only identified by mass and retention time. How do you know that they're the same thing without identifying them? So that's something I just wanted to talk about briefly. Again, this is work done by Rui Pinto at Imperial College. And what he's done is address the problem of matching different LCMS data sets together, these untargeted data sets. Here you see two examples, data set 1 on the left, data set 2 on the right. And this is retention time versus mass to charge ratio. And essentially what we're asking is, how do we tell if this feature here, ringed in red, is the same as this feature here, ringed in red? So what he's done is produce a very nice pipeline where we can use different aspects of the information we have access to, which is just retention time, mass to charge ratio, and intensity. We can do some thresholding on essentially sort of looking for matching features within a window in those two domains. We can then model the trends in those differences in the mass and retention time. So here's the retention time trend over the run. Here's the mass differences trend in the run, comparing the two different data sets. We can then calculate residuals, go through a period of a process of scoring and refining those thresholds in a very non-linear and flexible way, as you can see here. And finally come out with a set of features which are matched between two untargeted assays. And when we look and see how well we've done on that by looking at a set of identified or annotated features, we find in the study we've looked at, it's actually really, really good at matching them up between these two different data sets. Here's just a way of showing you how that works. So this is the original data set, and you can see if I flick backwards and forwards between the original and the match data set, you do lose several features, quite a few features, because they're not present in both of those studies, but you do retain plenty of features which do match. And then finally, I just wanted to mention that there are many more things you can do to apply to this problem. So again, another thing that Rui is working on is using the isotope relationships. So this is isotope ratios, and you can recognize the relationships of those, creating networks of adduct to adduct relationships based on mass differences, using that big network of different adduct relationships to improve on those matching trends that I was showing you in the previous slide. I just want to mention another aspect of integration, which is pathway analysis, and Cecilia is going to talk about this in a moment. But essentially, using pathways, you can put different biological molecules onto the same map, can't you? Because both proteins, genes, and metabolites, they all work together in biology, and a pathway is a good representation of that. So Cecilia will probably say a few words about that at the end of her talk. But in general, what are the prospects for integrating data? I think one of the big problems is that people are often not clear why they want to integrate their data. Is it so that you can get a better prediction, or is it so that you can get a better understanding of what's going on, for instance, with multi-omic data? And I think it's really important to be aware of what you're trying to get at when you do select a type of integration approach. It would be nice to see some standardization of the approaches out there. It's a bit of a wild west still at the moment, and I think that probably will continue for a while. But it's important to remember that depending on what your motivation, your goals are, you will need to select different methods, and those will be standardized in different ways. Annotation is a huge problem for integration as it is for anything else. But if you have annotation, then at least you'll have a better prospect of interpreting your, the biology. And I just want to make the point that deep learning approaches, while they seem really exciting and new and very powerful, one of their problems is they're a bit black box. So they're based on neural networks essentially, and it's very difficult often to interpret what in the model was responsible for a given behavior of the model. So for instance, why were certain genes and metabolites put together in some kind of predictive rule? It may be really lost inside that black box of a deep learning model. But there are explainable AI approaches out there as well, which can, I think, improve on this. So in conclusion, I hope I haven't gone on too long. I'd like to say that we do need computational metabolomics, and that there are many aspects. If you hear people talking about computational metabolomics as just metabolite identification, well, there's plenty of other things, if you remember that map I showed you at the beginning. And I think, there are increasing reasons for using computational methods in omics in general, and that's partly because standardized analytical methods are becoming more prevalent, so we're all doing things in a much more consistent way. but the data is increasing in amount and complexity. But thankfully, the data analytical methods, so the computational algorithms, are improving a lot as well. So there's a lot to look forward to. So thank you for listening to me. I'd just like to acknowledge the people that contributed to our work, so particularly Gonzalo, Graça, Rui Pinto, who I already mentioned, and Cecilia, who you'll hear from in a moment, and our wonderful collaborators throughout the world, and obviously our funders. Thank you. And I will pass over to Cecilia now.
So thanks so much, Tim, for a really nice, and super engaging talk with a great overview of computational metabolomics for our EMN community, and highlighting a few of your research projects that address the use of annotation for metabo annotator and data integration within your lab. So audience members, there are a few questions in the chat and also in the questions tab, so please keep them coming. I'll now transition to Cecilia Vida to further dive deeper into pathway analysis research within Professor Ebbels and Dr. Lay's group. Cecilia, the floor is yours.
Hi everyone. So first I'd like to start by saying a big thanks to the Metabolomics Society for giving me this opportunity to present. So today I'm going to be presenting first half of my presentation on some recently published work about pathway analysis in metabolomics, and specifically focusing on overrepresentation analysis. And then the final part of my talk will focus on our current and future work on single sample pathway analysis and data integration. So I'd like to just dive straight into pathway analysis itself. So I've grabbed this figure from this great review paper on pathway analysis. And essentially pathway analysis is a way to identify enriched or active pathways in an omics data set. So the inputs for pathway analysis are a data matrix, and normally these consist of samples and measurements of your data, such as metabolites or genes or proteins, and a pathway database such as KEGG and Reactome. And then we feed these inputs into a variety of different algorithms for pathway analysis. And these three boxes are kind of in the order of how the methods were established. So the oldest and probably most known method is the overrepresentation analysis, which is what we're going to focus on today. And this works by essentially identifying differentially expressed or abundant compounds. And then from these compounds, we look at, within each pathway, the number of differentially abundant compounds and those outside the pathway, and within the pathway, and then we use these numbers to assess the significance of each pathway. And then moving on from that, functional class scoring approaches were developed. And so instead of looking at just the differential abundant metabolites, they look at creating gene level or metabolite level statistics for every single entity in the data set, and then using these to calculate pathway significance. So GSEA is a method based on functional class scoring, which you might be familiar with. And then finally, the latest sort of generation of methods is the pathway topology methods. And essentially what these do is they integrate information about the topology of the pathway network and use this in combination with graph theory approaches to develop things like a pathway impact factor, which can contribute to the assessment of the pathway significance. So moving on to pathway analysis in metabolomics itself, we know that pathway analysis is widely used in many studies to identify the enriched or active pathways. And according to a recent benchmarking study, the most common method used is overrepresentation analysis, not only in metabolomics, but also still in the gene expression transcriptomics world, just probably due to its ease of use and the simplicity of the test itself. So the vast majority of pathway analysis methods have been developed for use with transcriptomics data, but we know that metabolomics data differs from transcriptomics data in several ways, which Tim has already touched on in his presentation. But the first, I would say, is the low coverage of the metabolome. So given a specific metabolomics assay, you're far less likely to be able to actually cover the whole metabolome of the species that you're looking at, as opposed to using a sequencing experiment and being able to cover a large amount of the genes and transcripts present in that organism. And the second thing is compound annotation uncertainty. So there's always going to be a few compounds which we're not entirely certain what they are. And this is not something that generally tends to happen in sequencing based experiments. So in our study, the main objective and aim of it was to actually understand how pathway analysis, and specifically overrepresentation analysis results, are affected by metabolomics data-specific factors, and then to provide some suggestions and best practice recommendations for those who want to use these approaches in metabolomics. So we investigated several input parameters and factors, and the ones I'm going to be talking about today are the effect of the background list, differential metabolite selection approaches, pathway database choice, metabolite misidentification, and assay chemical bias. Now, just to give you an overview of our methodology and the data sets that we used to do this, we aim to select 5 publicly available mass spectrometry untargeted metabolomics data sets from various organisms, biofluids, and different study types, as you can see in this table. And then we use these data sets to test different combinations of the input parameters or gradually degraded the data and to simulate factors such as metabolite misidentification. So now, just to focus more on the overrepresentation analysis itself and how it works, essentially it uses a right-tailed Fisher's exact test to calculate a p-value for each pathway. And the null hypothesis of this is that genes within the pathway are no more differentially expressed than those outside of the pathway. And so this diagram here can help us understand overrepresentation analysis a bit more. So what we've got here in the large white circle is the entire metabolome of the organism that you're profiling. So given a certain assay, it's quite unlikely that you're able to profile every single compound in it. So what we have is a subset of those compounds, which we refer to as big N, also known as the background set. So this covers some of the compounds in the metabolome that you can profile using your assay. Then we've got M. So M represents the compounds within the pathway that you're testing. So with ORA, we test one pathway at a time. And then we've got small n. And small n is represented by this dark blue circle, and that represents compounds of interest. So these compounds of interest could be, for example, differentially abundant metabolites. So those are the metabolites whose means differ significantly, for example, within your control case and control groups. And then finally, we've got K. And K represents the intersect here, as we can see, between the list of compounds of interest and the pathway. And what ORA is doing is essentially testing the significance of this intersect. This can also be represented as a 2x2 contingency table. So we count up the metabolites which are within the pathway, not in the pathway, those that are differentially abundant and non-differentially abundant, and then we can use the following formula to calculate the p-values for each pathway. So moving on to the first factor that we investigated, which was the background set. There are 2 different types of background sets which we define, and the first is the non-specific background set. Now this could be, for example, all compounds present in the KEGG database, and it's often the default option in many metabolomics pathway analysis tools. And this is as opposed to an assay-specific background set. So these could be all the compounds that are annotatable in your specific data set itself. So what we did is on this plot, we compared the p-values obtained using ORA when using an assay-specific background set, which is shown on the x-axis, and a non-specific background set, which is shown on the y-axis. And for this, we used all the compounds in the KEGG database. And we generated results using all 5 of our example data sets. And we can see their p-values plotted on here. And what we can see is that the p-values when using the non-specific background set tend to be far lower and more significant as opposed to using the assay-specific background set. But the problem with this is that most of these could be false positives, as it's very highly unlikely that given your assay, you'll be able to detect, for example, all 3,000 compounds present in the KEGG database. So it's important to consider this when selecting your background set. Secondly, we looked at a few other properties of background sets. And for example, we found that the size of the background set actually affects the power to detect significant pathways. So a larger background set will always give you higher power. And this is something important to consider if, for example, you've got an NMR assay which has 100 compounds. You have to think to yourself, is that going to give me sufficient power to actually run a good pathway analysis and get results? So moving on to the second factor that we investigated, we looked at how differential metabolites or differentially abundant metabolites can be selected to input to ORA. So this is the first step of ORA, the pre-filtering of the differential metabolites. And the way that we investigated this was that we took one of our data sets, which is shown in this figure, and for all 250 plus metabolites in that data set, we ran a t-test on each of them, which compared the significance in the differences in means of each compound in the case and control groups, and we ranked the metabolites from lowest to highest p-value. Then what we did was each metabolite was added one at a time to the list of differential metabolites of interest, and then we ran ORA. And on the y-axis, we can see that this is the number of pathways significant at a certain p-value cutoff. So we can see that there's an overarching trend that as we add more differential metabolites to this list, the number of significant pathways gradually increases to a global maximum. And then as we continue to add more of them, it gradually decreases until we've added all those metabolites to our list of metabolites of interest. And we've also got plotted on this graph some common p-value cutoffs that people might use. So in red, we've got Bonferroni cutoffs, and in black markers, we've got Benjamini-Hochberg false discovery rate cutoffs. So the main message behind this slide and behind this plot is that it's important to consider the amount of signal that you add when you filter this list of differential metabolites and the amount of noise as well. So it's important to consider this when selecting this parameter. Also, we found it's quite clear that just the addition of a single metabolite at a time can really affect the number of significant pathways. So it's quite important to carefully consider this parameter rather than just using one cutoff and going straight to the next step. Now, the next input parameter for ORA is the pathway database choice, so you'll have to choose this yourself. And we looked into three common pathway databases which you can access. So these are KEGG, Reactome, and HumanPsych, which is part of the BioPsych collection. And we looked at different aspects of these databases. So, for example, panel A here shows violin plots of the distribution of pathway sizes within each of these databases. And we can see that, for example, Reactome has smaller pathways on average than KEGG and HumanPsych, but it's got a larger range of pathway sizes in general. And so pathways will also differ in terms of their boundaries, so where pathways are cut off within the network, and the composition of pathways such as even simple and well-known pathways such as the TCA cycle can actually differ quite a bit in their compounds from database to database. And the second thing that we looked at was how databases actually change from release to release. So the way we did this was we obtained 4 years worth of the Reactome human pathway database, spanning the period from 2017 to 2020. And Reactome has 4 releases per year. And then we compared things such as the number of pathways, the number of unique compounds in that database, and the mean pathway size. And we found that there were quite large increases in the number of pathways and unique compounds over the years, with over 200 new pathways being added and 300 new compounds. So this begs the question, are pathway analysis results only valid at the time that you run them? And Also, it's important to consider that your results are only ever going to be as good as the knowledge base that you use. So it's very important to be trying to use an up-to-date knowledge base if possible. Moving on to a metabolomics specific factor, we wanted to understand here how metabolite misidentification can affect pathway analysis results. And so the way that we did this was that we had to simulate metabolite misidentification. And this is because we weren't able to ascertain the level of misidentification in our original data sets. So what we did is we selected different percentages of misidentification to simulate. So this figure shows 4%. And then for this percentage of compounds, we randomly swapped them with compounds that had an identical chemical formula or a similar molecular mass. And then we ran ORA. And to quantify the effects of the misidentification, we defined two new statistics, which are the pathway loss rate, and this is shown in the red bars, and the pathway gain rate shown in the blue bars. Now, essentially what these mean, so the pathway gain rate refers to the proportion of pathways which are gained simply as a result of misidentification. And likewise, the pathway loss rate refers to the proportion of pathways which are lost simply as a result of misidentification. And so what we can see in the figure is that all 5 of our data sets actually had a pathway loss rate and gain rate greater than 0 at just 4% simulated misidentification. So it's really important to consider that even at such a low level of misidentification, which is perhaps maybe a representative figure, you get some pathways lost and gained purely due to this fact. So it's very important to consider the effect of this artifact in your analysis results. And the final factor that I'm going to talk to you about is chemical assay bias and how this limits the pathways accessible in a data set. So we took one of our data sets, which was profiled by the Metabolon platform. And some of you may know that the Metabolon platform provides 4 different assay types in their profiling. And so what we did was we mapped onto the KEGG human metabolic network, which is shown here. We mapped the compounds profiled using each different assay in different colors onto this network map. And what we can see is it's quite obvious that even though this data set has quite a good coverage, certain areas of the pathway map, such as glycan biosynthesis and metabolism, lipid metabolism, and, for example, biosynthesis of secondary metabolites, have very poor coverage. And now the problem is that when you're running a test such as ORA, unless you have at least 1 compound, or 2 depending on the thresholds used, present within each pathway, you will never be able to test the significance of that pathway. So it's very important to remember that with any given assay, you're likely to be quite biased in the pathways that you can actually test. So to conclude this section of the presentation, I just want to give you our suggested recommendations for pathway analysis and suggested reporting guidelines. So do use a realistic background set and specify the compounds that form that background set. So an assay-specific background set is a good way to do this. Do use multiple pathway databases and compare the results that you get from those, and specify the database used and the release number of it. Always use multiple testing correction to select the metabolites of interest, and if possible, do use it on the pathway analysis results too. And do use an appropriate tool or test to perform the pathway analysis, and state explicitly which test you use and the version if possible. And finally, do use organism-specific pathways. Now this is just important because in some pathway databases, you'll have a reference pathway map. However, some of your results might not be specific to the organism under study. So it's always important to try and use an organism-specific pathway database if possible. Okay, so moving on to the second part of my presentation, we're just going to focus on single sample pathway analysis and its applications in metabolomics data. So single sample pathway analysis, or gene set analysis, refers to methods used to transform gene or metabolite level data to the pathway space, and it does this by assigning a pathway activity score to all pathways in each sample. Now, to give you a visual representation of how this works, It starts with an input data matrix. So we've got samples as rows and metabolites as columns. And what we do is we apply a single sample pathway analysis algorithm, and then this transforms this matrix into one that has the same samples as rows, but instead of metabolites, we've now got pathways which summarize the metabolite activities for each of these. And so there are several applications that we can then do with this pathway transform data. And these include something as simple as comparing pathway scores between multiple study groups. So for example, if you've got a cancer data set with multiple different subtypes, then you can compare the different pathways that are enriched between those groups, which is something that you can't currently do with conventional approaches such as ORA and GSEA. But you can also use the pathway level data for some interesting downstream machine learning analysis, such as pathway level clustering and classification. We can also use it for pathway-based data integration, visualization, and things like survival analysis. But again, most of these methods have been developed for gene expression data, and we want to find out how well do they actually translate to metabolomics data. Now, very quickly, for those of you who are interested in using these, I'm just going to summarize the different types that we have of methods. So there's dimensionality reduction based, so based on PCA or SVD. There are those based on GSEA, such as single sample GSEA, and those methods based on the Z-score. And so one area of my future work is to look at pathway-based integration of metabolomics data with other omics data types. And so typically multi-omics data sets are integrated at the molecular level, but pathway-based integration can actually provide several advantages, such as improved interpretability. So we can interpret the results in a pathway context, increase statistical power by merging measurements of several entities into a pathway, and increase parsimony as there are less pathways than individual molecules we actually have to interpret. And so single sample pathway analysis are one way that we can make this integration possible. So with that, I would just like to quickly thank my supervisors, Tim Ebbels and Rachel Lai, the Fabien Jourdain group in France, Jacob Bundy from Imperial, and the Wellcome Trust for my funding. So thank you so much for listening.
Thank you so much, Cecilia, for this engaging talk. I would now like to invite you and Tim to maybe turn on your camera if you prefer, so that we can start the Q&A session. Also, the floor is now open for questions. We already have quite some questions, so I would start with the ones as in the order that they appeared. So the first question is from James Matlock. This question is for Tim, and he would like to know what tools or online tools would you recommend for multiple assay integration and study integration?
Right, okay, that's a bit of a big question. There are, I would say, like I said, it's a little bit of a Wild West. So you have, although there are some tools out there which are specifically sort of sold as integrative omics modeling tools, I think it's best not to restrict yourself only to those. But to mention one or two, there's a package called Mix Omics, for instance, which is a nice package. I think it's an art package which has a multitude of different methods in it, mainly multivariate statistical methods. On the pathway side, there are things like Impala, for instance, which can integrate metabolomic and anything to do with genes, which is something that we contributed to in the past. I won't go into a sort of big long list because I'll bound to miss important tools out there. But I think the other thing to say is that it's good to think of what strategy you'd like to apply. So, you know, do you want to do, I don't know, multi-block PLS or something, or do you want to do a simple clustered correlation matrix or something like that, and then think, okay, well, how can I implement that? And it may well be that there are some generic statistical tools out there, things like Simca or you know, packages in R or Python that, you know, can affect that just as well as something that's supposedly dedicated to integrative analysis. And I should probably mention, you know, there's always commercial tools out there as well, which are specifically directed at omics analysis. So, you know, again, one can go down those routes if you've got the money to pay the license. I don't know, Cecilia, if you can think of any other tools that I might have forgotten.
No, I think that's a good summary. There's just one more called MUGSA, and that's like a multivariate integration of omics data, but they also project pathways onto it. So if you're interested in pathways as well, you could try looking into MUGSA.
Okay, thank you. Our next question is, in the case of the same, so this is for Tim, I think, in the case of the same cohort analyzed by different methodologies, for example, HELIC or reverse phase methods, how should the metabolites that are detected by both methods be treated? Should these duplicates be filtered before statistical analysis?
Very good question. And I think it doesn't just apply, you know, that question doesn't just apply to integrating multiple assays. But of course, even with one assay, you may have multiple peaks from the same metabolite. You may have, you know, 20 peaks from one metabolite and only 2 peaks from another metabolite. So there are differences in the representation of individual metabolites, which may well bias your statistical analysis or even a pathway analysis or something when you build that on top of it. How you should treat that is not something which is I don't think has a single answer. What people do in some areas, if you want to balance things, is you select what you might call the best representative variable for a given metabolite. But obviously, if you've already got you know, a table of metabolites and concentrations, then you can decide which of the measurements in which of the assays you feel is the best representation of that metabolite concentration and select that. If you're at the level of just looking at peaks that may be you know, identified by a mass to charge ratio or something, then it may be that you can select one variable from several that represent a given metabolite across assays using some statistical approach. So for instance, again, another approach that people are using is to select the one with the most significant association to an outcome, so the smallest p-value, for instance. But that, don't forget, may not be, although it sounds like a good statistical approach, it may not give you the variable which corresponds to the best analytical quality. So you may want to, for instance, go to quality control samples, or if you have in your experimental design dilution series, you could see which of the variables that metabolite best represents its concentration in terms of linearity, for instance, or reproducibility. So, as always, there are many different answers. I think what most people do, at least initially, is don't make that selection, because what most people are trying to do at the beginning of an integrative analysis is to explore the data and see what exists on different assay. You may not even have identified metabolites for all, you know, in all your assays. So usually people leave all that information in to begin with, and then perhaps once you've decided on, you know, exactly the sort of final analysis you want to do, and you're aware of all the strengths and weaknesses of the data that you have, then you can do this sort of single variable selection. And I suppose I should also mention that, you know, corresponding to what people do in the gene expression world as well, there are ways of summarizing a metabolite by combining multiple variables together. But again, there's no standard way of doing that. I mean, you could think of just taking an average, for instance, but obviously what you would like to do is combine variables which have the best sort of signal to noise, if you like. You don't want to include noisy variables. So again, that area is not well developed at all. And ultimately, a lot of it comes back to this problem of identification. If we knew which metabolite every variable was corresponding to, then it would be a lot easier for people to do this kind of selection. Still wouldn't be easy, but it would be more straightforward. If you have in your assay plenty of unidentified variables, unidentified features, then the question is what should you do with those, and should you be combining those as well in some way? So it's hard to know. Right, so again, another sort of not black or white answer, I'm afraid, but there are things you can do in terms of variable selection.
Great, thank you. There's yet another question for you. First, so this question is from Justin van der Hooft, who would like to thank you for providing such a great overview of the field. His question is, what do you think are the main that lie ahead of us? And what is the most exciting thing that you would like to work in the next 5 years?
Wow, sounds like an interview question. Thanks, Justin. It's really nice to hear from you. I'm glad you could make the talk. So where do we think this field is going, I suppose? Well, don't forget, it's not disconnected from the broader field of metabolomics itself. So one aspect is the computational methods to some extent follow the data that's being produced. So for instance, ion mobility spectrometry is something that's come to the fore in the last sort of 5 or 10 years, and there's plenty of algorithms to deal with that kind of data which would have not been there in the past. So it may be that there are approaches coming along in the analytical sphere, in the instrumentation sphere, where we'll need to develop new methods computational methods to deal with those. So I don't know, perhaps it's hyperpolarized NMR or something like that, or more sophisticated types of multidimensional mass spectrometry or something. Obviously, I would be silly not to mention deep learning approaches, because they've taken over the whole world of machine learning and AI. And I think there is a strong need to investigate how those can be applied, and people are already doing that, of course. And over the last few years, there's been lots of papers in metabolomics using these approaches, but perhaps not as many as you might have expected, given how much they've sort of had a big impact on other areas of science. And I think one reason, as I mentioned, is this black box problem that in metabolomics and omics in general, we want to do the modeling, statistical modeling, but we also want to know why it works. And if the machine learning approach that you are applying does not tell you easily which are the important variables or how they contribute to the prediction that you're making, then the approach is less useful. That's not to say that those approaches can never be used. It really depends on the application. So, if you have something where you're simply looking for a classification, you don't really care how it's made as long as it's accurate, then those approaches are fine. But I think, as I mentioned earlier on, in deep learning and AI, there is a lot more emphasis in the last couple of years on explainable AI. And so how can you get an understanding of how the machine is working? Other problems with deep learning, of course, is that it requires a huge amount of data for these complex networks to train. And typically in metabolomics, although we may have a lot of data, we often don't have a lot of labeled data. So if you're thinking about the problem of metabolite identification, we might have loads and loads of spectra, but we don't know what all the compounds in those spectra are. And so the number of spectra for pure compounds in metabolite databases might be relatively low, where I'm talking a few thousand is relatively low. But there's plenty of opportunity for those techniques to come to the fore, especially things like image analysis techniques, where deep learning's made a big impact on image analysis generally. And of course, our data in metabolomics can often be represented as an image, so a two-dimensional NMR spectrum or a multidimensional fragmentation approach in mass spectrometry or something. So kind of a long-winded answer, I suppose, but I think there's a lot of different areas. I think we will see impact of deep learning, but I don't personally think that it's going to be the panacea that maybe people might expect from its impact elsewhere. And I think, ultimately, as I said before, so much of this comes back down to metabolite identification. Although it's not the only problem in computational metabolomics, lots of other problems depend on getting it at least some level of correct. But of course, I don't think we're ever expecting to identify all metabolites in all data sets. So we're going to have algorithms that allow you to do what you want to do without having complete identification. So progress on identification will be a key point in the next 5 years, but we need to be able to develop algorithms that, for instance, can do this matching of data sets I was showing you early on without having that knowledge, sort of complete knowledge of the IDs. Okay, I better stop talking there, I think.
That was very informative indeed. The next couple of questions are for Cecilia. So the first question is, what about the pathway definitions? It is rather arbitrary where pathway starts and where it ends. For example, if specific reactions belong to this or that pathway. Did you check the effect?
So it's not something that we looked into specifically, but it's definitely very, very important to take into account, because obviously your pathway results will depend on what the definition is in each database. So what I would suggest is just to use several different databases, and then just compare the results that you get. And also look into the definitions of the significant pathways, look into comparing whether they're different from database to database. You can also supply your own custom definitions when using these approaches. But yeah, hopefully as the knowledge bases continue to evolve, then those definitions will become more and more reliable and useful to us.
Okay, our next question is from Stefania, who first likes to thank you for your talk and says that I'm particularly interested on the figure that showed how the number of pathways increased with increasing number of features added to the analysis. Any advice or tips on how to choose the number of features we should add into the pathway analysis?
Yeah, that's a really great question. And what I would suggest you do is maybe experiment with a number of different cutoffs, multiple testing correction methods, and you could also plot a similar figure yourself if you wanted to. So all our source code for that piece of research is available on my GitHub. So if you wanted to reproduce the figure, then you could go ahead and maybe try that and see what it looks like on your specific data set. Obviously, we don't want to encourage, things like p-value hacking. So for you to select exactly the point on that graph where you get the most significant pathways. So there's no definitive answer, but I would just really encourage you to experiment with different things.
I think I would just add that part of that figure that we're talking about, where the number of pathways goes up and then down again, is you can see how unstable it is. The number changes very significantly, even adding one metabolite to the list, as Cecilia showed. So I think if you're ultimately, when you do the pathway analysis, what you're trying to build is a sort of biological story, and you don't want that story to be dependent on whether one metabolite gets added or not, right? So you want it to be consistent and stable. So I think this plot that Cecilia showed is a really useful way of visualizing whether the story that you want to tell from the list of significant pathways is somehow reliable. If you get a relatively smooth area of it, then perhaps that is a piece of evidence that is indicating that the story is fairly consistent across this range of thresholds. But it's still hard to select a specific threshold because, of course, it's an arbitrary selection anyway.
Yeah, indeed, a very interesting plot. I've also tried that with the data that we are generating in our lab. Okay, moving on to the next question. It's from Julie. How do you address and practice the impact of misidentification? We obviously have difficulties knowing the extent of it in our data sets. What can we do apart from mentioning it in limitations of the analysis?
That's a really good question. I don't have a definitive answer for that. I don't know if Tim maybe can help elaborate a bit.
Well, I think there are things you can do Whether it's easy to do them or not is another question. Obviously, Cecilia did something a little bit along these lines in her research. So you could, for instance, take your results and then say, okay, what happens if I swap a small number of metabolite IDs for randomly chosen ones? Do I still get the same biological story out? Or even going back further in your pipeline, you know, what happens if I add noise to the data that I obtained? You know, would that change the story? It's a little bit along the lines that I was talking into the previous question. You know, how stable is your analysis given the data that you have? Is it robust to, you know, changes in assumptions or equivalently noise in the data? And noise in metabolite ID is a very relevant thing. Now, what the level of misidentification in data sets in metabolomics is, we just don't know. There are some estimates out there, but I don't think they're comprehensive. And depending who you talk to, you know, they'll say, oh, it's, you know, very, very, very tiny, or some people will say it's actually quite a lot. So it really depends, you know, the methods that you've used and stuff. So it's hard to actually know. And I think when it's your own data, of course, you're even less keen to believe that there are misidentifications in there, right? But you've got to remember, if you've used especially a computational identification or annotation technique, it's likely that there will be some misidentifications. Or when we talk about misidentifications, of course, we don't necessarily mean something that is completely wrong. It could be that you have an ID which is too specific, and the data that you have only supports really an ID to a sort of a higher level of hierarchy. So I might be talking about, you know, a class of metabolites. You can definitely identify the class, but the ID you're using in your software is more specific than that, a particular molecule within that class. Yet the data you've got can't really say that for sure. And the reason that you've got this more specific ID is, you know, a sort of foible of the approach that's being used. So those types of uncertainties are quite tricky to understand and work out what impact they'll have. But with computational techniques, of course, it's relatively easy to swap things in and out, add randomness and stuff, and see how it does affect things. So I would recommend that if you can do that without too much difficulty, then give it a go. Even something simple as, you know, you're doing a pathway analysis, you've got a web-based tool where you plug in some metabolite names to see what pathways you get out. Just try swapping through those names. You know, that's an easy thing to do.
That's indeed very helpful. Our next question is for Cecilia again. Could you elaborate more on the pathway-based data integration? Do you have some example?
So I can say that what we're looking into doing is essentially trying to integrate metabolomics with, say, transcriptomics and proteomics, and specifically integrating data sets that have the same samples, but they'll have different features, of course, different metabolites, different genes and proteins. And then essentially what we're looking to do with that is to use some kind of integrative method, such as the ones that Tim was mentioning, like the mixomics. So those will integrate the different data sets, and they'll provide you with, for example, different factors that represent what's going on in the data, and these factors are composed of genes, metabolites, and proteins, which are significantly contributing to that factor. But we want to build upon these kind of techniques to do it at the pathway level. And there's two things that we can kind of achieve by doing that. One is to understand the significant pathways across the 3 omics, or number of omics that you're integrating. So we've got pathways that are significantly enriched or depleted across all 3 omics, and hopefully we can do that in such a way that biology underlying economics data kind of makes sense, how the pathways will interact across omics. And the other advantage of this is by integrating things at pathway level, and using these integrative methods, we can also do things such as multi-omics pathway-based classification. And you might find that in some cases, the classification performance might actually improve by doing it based on pathways, rather than individual molecules themselves. So these are the two main outcomes that we're looking to achieve by doing this.
And maybe I can just add a small thing to that, which is we're finding in Cecilia's work particularly that, you know, there are major differences between doing pathway analysis with transcriptomic data, which is where it's most traditionally done, and in metabolomics. And those are mostly or largely due to the coverage. So metabolites coverage is much, much smaller. You may only have 3 metabolites in a pathway, whereas perhaps you might have had 20 or 30 genes. And so we've found that, you know, simply using the methods that are available in transcriptomics and elsewhere in bioinformatics and applying them to metabolite data is not straightforward, and also potentially can be dangerous. You know, you might end up with more dangerous results. So I thought I would just bring that up because it is something that's sometimes quite easy to do. It makes Cecilia's life much harder, unfortunately, but, you know, that's what we're trying to look at, to see how, you know, which methods can be used reliably, and how can you compensate for this massive difference in coverage between the different omics.
Okay, thank you. Our next question says, which online server which online server would you use for metabolomics, just like we use Genome, GNOME AD, Predict SNP, Haddock, and PDB-Sum for DNA and protein.
Okay, so I would say the most well-known is the Metabolomics web server. So it's really good. It's very comprehensive, and it does so many different things. So you can do things from like metabolite ID conversion to PLS to pathway analysis. So I would suggest looking into that. However, it's important to really try and understand what each function on that server is actually doing. And when it gives you the opportunity to tune the input parameters, so say you're doing pathway analysis and you're specifying a background set, then do take advantage of those to try and make the most of the results and get them as sort of robust and reliable as you can. There's also an R version of Metabolomics as well, if you wanted to use the same on R. But yeah, I think that's the main one that's available at the moment.
Thanks, Cecilia. Also, the next question is for you again. It's from Yash, and he says that I'm doing metabolic work, particularly with pathways with algae. I am currently using BioSeq. Are there any other online tools or resources that you can suggest for algae related work?
Sorry, do you mind repeating the last word? Can you suggest?
Algae.
Oh, algae. Okay. I'm going to be 100% honest. I'm not 100% sure of specific tools for that. But yeah, do look into Metabolomics. Yeah, that's what I would suggest.
Cecilia, you have worked with BioSeq a bit. Do BioSeq have their own pathway analysis tools on there? Because I know they have databases, but do they have the analysis tools?
I'm not sure about BioSeq. I know that Reactome does have it, but I'm not sure if they have the algae species on there. But yeah, have a look into BioSeq because they have a lot of databases for a lot of different organisms and things, so that might be helpful as well.
Okay, our next question is for Tim. So correlation-based approaches give a lot of false positive pairs, specifically in terms of integrating transcriptomics and metabolomics. Could you tell us if there are other options available besides correlation-based approach for this type of integration?
Okay, so I think firstly it depends how you define a false positive. You know, if you mean two variables which appear to be correlated, but that doesn't represent a reliable statistical relationship, perhaps because there's an outlier or a strange distribution or something, that's one thing. And those types of problems can be filtered out, obviously, with things like outlier detection. Then there are things that you might consider false positives because they're actually indirect correlations. So, you know, if A is a cause of both B and C, where B is a metabolite and C is a gene, for instance, then B and C will be correlated to each other when that doesn't actually represent a causative relationship between B and C. And, you know, one can use techniques like partial correlation to try and remove those types of association. So if you look in the literature, there's plenty of techniques and packages to use partial correlations using so-called Gaussian graphical models to look at partial correlation networks, basically, which are usually much sparser than the full correlation networks you might find. Of course, there's non-parametric approaches, so things like Spearman and Ebbels correlations, which can help with the outlier problem I mentioned. And then if you want to sort of go sort of to a higher level, as it were, then there are multivariate approaches, of course. So if you look at either things like multi-block chemometric methods, so things like hierarchical PCA or multi-block PLS, those approaches are trying to take account of not just simple two-way correlation, but the entire sort of complex correlation structure within one block of data, but also across different blocks. And of course, different methods will model that in slightly different ways. So methods such as orthogonal PLS, for instance, can be used to filter out spurious variation, for instance, that is not related to the effect of interest, for instance. So there are some quite sophisticated methods there that, while they're still based on correlation as an overall concept, it's a more refined and sort of high-dimensional version of that, which should allow you to pull out the correlations which actually are more reliable and hopefully more important. But at the end of the day, that's still correlation, doesn't tell you whether something is causative. And, you know, how to integrate data without using correlation at all, well, you know, the pathway approaches that Cecilia was talking about is one way to do that, although even there, you expect to see a lot of correlation in the data, but you may not need to use it to tell a story. Beyond that, I think it really comes down to it depends on your experimental design. You know, if you have a time series where you can see the effect of an intervention over time, then you may not call it correlation, but an association between the treatment and time gives you some idea that there's a relationship between you know, the intervention and the outcome. And if you see that in both, you know, say a set of genes and a set of metabolites, then, you know, you can integrate data that way. But I would say that's still really a kind of correlation. So it's, I don't want to say that you can't ever get away from correlations, but I think it is the, you know, simple correlation. Yes, you can do a lot more than that, but at the end of the day, you're always going to be looking at some kind of statistical relationship between variables, and you know, that is a type of correlation, really. So perhaps I would stop there.
Yeah, thank you. In the view of time, I would just take one last question. It's also quite an interesting question. So sometimes a compound is so strongly differentially abundant that one pathway will seem very significant, even though there is only this one compound belonging to it. Would you give more credit to a significantly impacted pathway because several compounds in it are differentially abundant, but maybe to a lesser extent? I think this is for Cecilia.
I think I would give them equal credit, just because even if one metabolite is very, very strongly differentially abundant and it's causing the whole pathway to be enriched, although the others might not be also that significant, it's still, you know, it's still likely that in that system, those pathways containing that metabolite are still affected just because of its very, very high abundance. differential abundance. So yeah, I don't think I would place more emphasis on the case where you've got just one metabolite differentially abundant than the case where you have several working together in effect. I think they're both important cases.
Yeah, I would really agree with that. I think, you know, if you look at metabolic modeling and you look at how different metabolites in the same pathway may be affected by an intervention or change, some metabolites may not show any difference, even if the pathway is really affected, and other metabolites may respond a lot. There also, don't forget, may be metabolites that are changing, but you cannot detect them, or you have measured them, but for some reason they're not able to be mapped in the pathway analysis. So there are plenty of ways in which you can lose metabolites in the sort of pipeline, as it were. So I know it's conceptually makes a lot of sense to differentiate those two situations, but I think in practice it's not necessarily as straightforward as that. And it also, at the end of the day, you have to think, well, what does it really mean for my biology if I see one metabolite with a huge change? Maybe that is important for you. Perhaps in metabolic engineering applications, it might be your product or something. But personally, we are trying to look at these. The reason we're looking at pathways is because we expect there to be a coordinated change in a whole set of metabolites or a set of genes. So we certainly would not like it to be the case that the analyses all favor pathways where really only one metabolite is changing. I think we are looking for these patterns of change across across the pathway.
Great, thank you. With this, I would like to wrap up this webinar. And firstly, I would like to thank you, Tim and Cecilia, for such nice talks, and also for your time to answer these all the questions from our attendees. And also, I would like to thank all the attendees for attending this EMN webinar session. More details on the next webinar would follow, so please stay connected on our social media channels for that. Thank you.
Thank you. It's been very nice. Enjoyed it.
Thank you.